Hello, everyone. Welcome to today's live broadcast, Improving Confidence and Productivity in Released Glycan Analysis for Biotherapeutic Development. I'm Rita Peters, Editorial Director of BioPharm International, and I'll be the moderator for today's event. We're pleased to bring you this webcast presented by LCGC and BioPharm International magazines and sponsored by Waters Corporation. I'd like to share a message from our sponsor. With 60 years of research and development of specialty measurement instrumentation, Waters' LC-MS, chemistries, informatics, and thermal analysis innovations help pharmaceutical and biopharmaceutical companies efficiently accelerate and improve workflows. The company's deep understanding of the global pharmaceutical industry, combined with its advanced analytical technologies, help bring critical medicines to patients quickly, safely, and effectively. I have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of your screen. You can enlarge the slide window by clicking on the small square icon in the upper right-hand corner of the slide window, or you can hover your mouse over the lower right-hand corner and drag the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing the presentation, please click on the question mark help widget in the dock at the bottom of your presentation window. Now I'd like to welcome today's speaker. Shimo Zhang is a senior scientist in the biopharmaceutical group of scientific operations at Waters Corporation, where she applies her expertise in separation science and mass spectrometry to support the development of analytical instrumentation and biopharmaceutical marketing initiatives. Recently, her focus is on the development of high-throughput screening methods for released glycan analysis using the BioAccord system. So thank you for joining us today, Shimo. I'm going to turn the presentation over to you.
All right, thank you for the introduction, and thank everyone for joining us today. So today I'll be talking about recently developed workflow for released glycan analysis and how it can be used to improve the productivity and confidence in biotherapeutic development. In this presentation, I will start with a brief introduction on the challenges of glycan analysis. Then I'll introduce the workflow we developed from sample preparation to data acquisition and data interpretation. And at the end, I will talk about how this can be transferred to a screening workflow for high-throughput glycan monitoring. As you know, glycosylation is the carbohydrates, what we call glycans, attached to another molecule. In this case, it's the monoclonal antibody. The glycan on the Fc region usually is about 2 to 3% of the total mass of the IgG, and sometimes you can see the N-glycans also linked to the Fab region. The glycosylation usually is sensitive to manufacturing conditions and has impact on many properties of the drugs, such as the effector functions, ADCC, and half-life, anti-inflammatory, and also immunogenic response. So the glycosylation is commonly assessed through released glycans, where we can determine the critical analytical specs such as identity and relative abundance of the released glycans, and also the consistency across batches, as well as some specific glycan attributes, such as listed on the left side of this slide. However, there were some major challenges in released glycan analysis. Since glycan is not optically active in their native state and also difficult to ionize in the MS analysis, the sample labeling is usually required to increase the fluorescence and MS response. There are conventional sample preparation methods, which is time-consuming and often involves many steps. MS sensitivity is often still an issue. And on top of that, data analysis can also be frustrating, including there are lots of manual steps for glycan identification, especially for closely eluded species. So therefore, a simple and automated workflow can be very helpful in released glycan analysis. To improve the productivity in released glycan analysis, we developed a simplified workflow, including 3 steps. Specifically, the RapidFluor MS labeling technology, when combined with the water separation columns, enables the use of innovative technologies, such as the BioAccord system, and all of which can be controlled through the unified software for data acquisition, processing, and reporting in a compliant-ready environment. Within this workflow, everything needed was provided in one easy kit or platform. So next, I'll discuss this workflow and look into more details. Starting from the sample preparation, the RapidFluor MS glycan labeling kit were designed to reduce the preparation time and also increase the MS sensitivity of the glycan samples. Now let's take a look at the glycan labeling method first. So as we know, after glycan being removed from the protein molecule by PNGSF, and it has a reducing end called glycosylamine, which cannot be directly reacted with the conventional labeling reagent such as 2AB. And these glycans have to go through additional reductive amination steps to produce free glycan in order to react with 2AB labeling reagent. And this reductive amination step is lengthy and requires high temperature. And in addition, it also requires the transfer from aqueous to anhydrous reaction condition, and this leads to additional cleaning and drying steps. Moreover, the amine reaction also produces heterogeneous reaction products, as it's possible to have multiple labeling reagents connected to the same glycan, and this may lead to additional problems in the following analysis. On the other hand, the RapidFluor MS labeling method takes a different approach. So as shown on this picture, the RFMS labeling reagent contains 3 groups, including one rapid NHS carbamate rapid tagging group, and a fluorophore group, and a tertiary amine group for enhanced MS sensitivity. And this NHS carbamate rapid tagging group can directly react with the glycosylamine reducing end, and so that the glycans don't need to go through additional reaction steps. And in addition, this reaction rate is very fast, which allows the reaction to be completed within seconds. So this RFMS labeling reagent, after reaction, can produce a very stable and highly specific linkage called urea linkage. And therefore, the reaction product is very specific, so you don't have any heterogeneity in the sample. And right now, this RFMS labeling reagent was introduced in the kit. So here is an overview of how to use the RFMS glycan labeling kit to do the glycan sample preparation. So the first step is deglycosylation. And this step is to unfold the protein to let the glycosylation site be exposed to PNGSF for glycan removal. And if we are using conventional labeling method, this process will take more than 16 hours because we have to do pre-labeling cleanup and additional drying steps. So with the glycan RapidFluor glycan labeling kit, there's a new enzyme being introduced in this kit, and it's called RapidJest. So this RapidJest is a surfactant. It can denature the protein while still keeping it soluble in aqueous condition. And in combined with the PNGSF glycan releasing, and this process only takes less than 15 minutes, which is a big improvement compared to the lengthy reaction with the conventional method. And the next step is the labeling, which is using the RFMS reagent we just introduced. And then the final step is cleanup. So here in this step, we use the SPE plate-based technology to purify the glycans from the reaction. And in this step, all the byproducts, such as the labeled proteins or double labeling, anything will be cleaned out, and you will have the pure labeled glycans and followed by the FLR or FLR-MS analysis. And this sample preparation, the whole process, you can do it within 1 hour, which is a remarkable improvement compared to the 24-hour preparation with the 2AB labeling method. And this improvement not only enhances productivity on the sample preparation side, but also on the detection side, thereby increasing, certainly especially with the overlapping peaks, and also reducing the amount of follow-on analysis that may be needed. And on this slide, it shows the comparison of RapiFluor-MS labeling glycans against some other commonly used labeling reagents. And so here it shows for both FLR and MS, the RapiFluor labeled glycans had a much stronger signal compared to others. Versus instant AB, they were 2 times higher for fluorescent response and about 1,000 times higher for MS response. And similar trends can also be seen for the other 2 labeling reagents. And we can also notice that the balanced signal was obtained for FLR and MS, meaning that any peak you find in the FLR chromatogram, you can probably find the corresponding information in the MS as well for confirmation. To further improve the productivity, the sample prep can be partially or fully automated using the existing RFMS labeling kit. This kit is validated, so there were 2 options for you to use for automation. One is semi-automation with Andrew Alliance. This is for lower throughput and cost automation, and there are other automation methods such as Hamilton or Tecan. This is for higher throughput automation. And all of them, we have automation scripts available when you purchase the instrument. So in this way, we can make sure the process is scalable and deployable. The next part in the workflow is to increase productivity with the robust separation and detection technology. Specifically, here we use the recently launched BioQuer system. The BioQuer system was introduced by Waters in January this year, configured with the Acuity iClass Plus UPLC, Acuity RDA TOF mass detector, and also unified control software. This is a benchtop system with great performance for biopharmaceutical analysis. The system was specifically designed to be easy to use, robust, but still with high performance, an automatic system setup and built-in self-diagnostic tool. As you can see from the front panel, this is what we call Smart MS. It is for the users to troubleshoot the system more easily and be able to get the users to resolve the issues by themselves. On the instrument front panel, the color-coded symbols represent different system conditions and possible issues, which simplifies the troubleshooting steps and also reduces the instrument downtime. As one of the 3 supported workflows on the system, released glycan analysis can be performed in a simple and efficient manner. The Acuity RDA mass detector setup page here showing the Smart MS-enabled auto-calibration function for efficient instrument operation. With only 1 click, the instrument can be calibrated for desired polarity and mass range in an automated fashion. The instrument method setup was also simplified. So here shows the instrument method setup page for MS data acquisition. Here shows the parameters that can be adjusted. For released glycan analysis, the low MS range option was selected, which is 50 to 2000 mOV. And the last but not least step, which is the informatics. In Unify, there's a workflow specific designed for released glycans, which allows automated data processing using the query retention time and accurate mass information of separate glycans. In the workflow, retention time of glycans were calibrated against the dextran ladder and converted to glucose unit values, and then used along with the accurate mass information to conduct a library search within Unify for peak identification. And now just a little bit background on the GU library. In this library, the GU values were experimentally determined for each glycan. By using different enzymes to cleave off different monosaccharides and separating the result glycans under the same platform method, then calibrate against the dextran ladder. Both GU and glycan structure can be confirmed and added to the library. So currently, there are 168 glycans in the RFMS library and more in the 2AB and 2AA library. The GU values significantly improve the robustness of data reporting. So here shows the round-robin study that we did to evaluate the GU values for the improvement in reproducibility. So now the multiple sites reported released glycan helix profiles in terms of retention times, and there were significant deviations were found in the dataset. As you can see from the figure in the left, the percent RSD value is about 8.5%. Upon conversion from retention times to GU values, the RSDs were reduced to less than 0.5% with the GU calibration. In addition to improved accuracy and precision of data reporting, the GU value can likewise be used to make glycan assignments less ambiguous. So if you think about the glycan workflow we talked about previously, with the use of GU value for library search, the highest robustness of peak assignment can be ensured by minimizing the potential variation from retention times across analyses, while the use of mass confirmation also adds extra confidence to the assignment. And here shows the repeatability test using 20 consecutive injections of RFMS glycan standard. There are 3 glycans were selected to test the repeatability of the system throughout the day by using relative abundance as a measure. The relative abundance was calculated based on the ratio of the selected peak area and total peak area. The results here showed high consistency for relative abundance across the 20 injections, which demonstrated a high repeatability of the workflow. To demonstrate the released glycan workflow, here we show the example of the Orencia, which is epidercept. It's a fusion protein that contains a complex mixture of glycans with a high level of acylation. As you can see in the structure, it has 6 glycosylation sites. And now with the mass load of 4 picomolar, the processed FLR trace was shown here, with each peak automatically assigned and also annotated with its glycan name and associated structure information. And these are the information that you can find in the glycan structural library in Unit 5. And in addition, we can also see the good MS signal response in the TSC chromatogram below here. This demonstrated the balanced FLR and MS sensitivity of the labeled glycans. So with the RDA detector, the high-quality MS full scan and XIC data can be also obtained for critical glycan species. In this example, we're showing the Manos-5, which has only a relative abundance of 0.2%. And in this chromatogram, it shows the high signal-to-noise response for the XIC, and below here shows the full-scan MS spectra. This high sensitivity afforded by the RDA detector resulted in the 31 unlinked glycans being identified within a 10 ppm mass tolerance. Supporting analysis information such as mass error, GU values, expected and observed mass can also be accessed through this component summary table. The view of the results in released glycan workflow can also be customized to fit into the purpose of different experiments. For example, to support the comparability test in process formulation change or biosimilar development, the workflow steps are clickable and can be customized and directly lead to the data for straightforward comparison of the results, so that the efficiency can be maximized in the product development. Here, we use infliximab as a case study to demonstrate its use in biosimilar development. So infliximab is a mAb-based drug to treat for arthritis. It has 2 glycosylation sites on the Fc domain, and it has about 25-ish glycoforms. With the Rabiflor MS labeled N-glycans, after they were released from innovator and biosimilar infliximab, they were analyzed by helix separation, then followed by the inline FLR and MS detection using the BioAccord system. The process, the FLR traced the innovator infliximab with each peak assigned and annotated with the glycan name and structure information were listed here. With the assigned glycan structures, the similarity of the glycan profiles can be then assessed by performing relative quantitation for all identified glycans and then binary comparison between the innovator and biosimilar drug products. Within this workflow, a direct comparison was made by overlaying the 3 FLR chromatograms of the innovator and biosimilar mAb. So here in this chromatogram, the black trace and the red trace represents the 2 samples from innovator infliximab, while the blue trace was from the biosimilar mAb. The overlaid chromatogram of the 2 innovator and 1 biosimilar sample show the high degree of similarity of the glycan profiles between the innovators, and then the differences were found in relative abundance for multiple low-abundant glycan species between the innovator and biosimilar mAbs, as shown in this zoom-in chromatogram. So to confirm these differences, a summary plot can be used to quantitatively compare the FLR response or relative abundance of selected glycan species. As shown in the summary plot of percent amount on the right side, we can see a consistent relative abundance of FA2G2 SG1 in the 2 samples in the innovator mAb, and also a much higher abundance of this glycan in the biosimilar mAb. When performing release glycan analysis of biosimilar mAbs, it is quite common to come across glycans with a challenging MS spectrum for peak identification, such as isobaric glycans, and this might lead to inconsistency in glycan analysis results. The high-energy fragmentation is a common MS-based method to help differentiating isobaric glycans with similar structures. And in this case, the RDA detector has the ability to acquire low-energy and high-energy MS fragmentation spectra at the same time, which provides additional diagnostic fragmentation ions to improve the confidence in structural assignment. As shown in this chromatogram, the highly similar GU values cause that a glycan FA2G2 closely eluted together with its immunogenic isomer, which is FA2G1GA1, as shown in this XSC. So as shown on the MS spectra on the right side, the high-quality MS full scan and fragmentation were obtained for both peaks, while diagnostic ion, which is M/Z528, for alpha galactose was observed in the MS fragmentation data of the shoulder peak. But this confirms the identity of this peak is FA2G1GA1. To further accelerate the glycan analysis, the Unify can automatically generate analysis reports to simplify the workflow, and so here we show the review, sharing, and filing of acquired data can be done through this reporting function. To demonstrate this function, a report was created using the customizable template to summarize the glycan analysis results. As shown in this slide, the generated report was formatted to include analysis information, profile summary, chromatograms, and also component summary. And this shows the direct comparison of the relative abundance of different glycan attributes across samples. And this is a unique feature of this release glycan workflow. So it does the calculation automatically and doesn't require any manual input. And also to meet different requirements in laboratories, the report can also be customized to include other desired information for simplified review of the analysis results. The analysis results, such as identified glycans, can also be transferred to downstream for monitoring use. So with the assigned glycans in the released glycan workflow, database containing accurate mass and retention time information can be built for individual type of samples. So if you have a sample like infliximab, you can characterize the sample and then build the whole profile of glycans for this infliximab, and then use that as your database to create a screening workflow. And therefore, the identification of glycans can be done based on their accurate mass and possible retention time, so that there will be no need to calibrate retention time, and therefore there will be also no need to run the detection ledger and saving extra time. In this screening workflow, you can also use different grading conditions and set up acceptance criteria for critical glycan species, so that the monitoring glycans can be completed in a more flexible and efficient manner. Here, we continue to use the case study of infliximab to demonstrate the transfer from released glycan workflow to the screening workflow. So similarly, with the sixth batch of infliximab, including 2 batches of biosimilar mAb and also 4 batches of innovator mAb, and the glycans from these infliximabs were released and labeled following by the procedures we discussed earlier with the rapid fluor MS glycan labeling kits. And be sure to note that one of the innovators was backed in with the high mannose samples in order to see if this can be captured in the screening workflow. And to increase the throughput, a 5-minute method was developed for the separation of these glycans. And based on the profiling results from released glycan workflow, a database was created for the target glycan species, which is about 20 to 25 glycans. And in addition, the acceptance criteria was also set up for the percent amount of mannose and also silated glycans. And here shows the results from the view of the sample plate. As shown in this picture, each column represents 6 replicates of the same sample from the same batch of the infliximab. The color-coded sample values represent the screening results based on the acceptance criteria. And by a brief look, we can determine that most of the results are within the spec, as they are showing as green. But the batch with the spec-in mannose sample was completed with error, meaning that this sample was out of spec. And additionally, you can see there's a column next to this one is shown in suspiciously yellow color, and that means it's still within the spec, but it's completed with warnings. So if we look into this result more carefully, we can pull out the summary plot just like what we did in the released glycan workflow. And now you can see the percent amount of the mannose 5 sample was much higher in this batch than others, which was expected due to the spec-in mannose 5 samples. And for the batch of innovator 2, which is the 6th column, we can see the percent amount of silated glycan species was within the spec, but was much higher than the rest of other batches. And this means there may be some change in this manufacturing conditions of this sample, and also maybe there was some sort of change in the instrument conditions. So this complete with warning can remind you to look into other things to confirm the results are acceptable. And similarly to release glycan workflow, the report can be generated to include key information. And here we show the color-coded results table and also the summary plots. And these color-coded results helps us to identify the out-of-spec results very quickly in order to determine the next steps for action. And to summarize, the glycan workflow we introduced today was enabled by the breakthrough technology RFMS labeling kit. It has a rapid tagging capability and enhance the sensitivity of fluorescence and also MS ionization. It is also automation friendly, which can improve the productivity even further. And in addition, we also use the innovative detection and separation technology, which is BioAccord system. It has robust performance with high throughput ability, and you can calibrate the system with only one click. And it requires minimum training and low maintenance, and also supported by qualification. And at the end, we have the ready-to-use workflows, which is release glycan workflow and also screening workflow within the unified software. It is automated and can be done within a compliant-ready environment. And together, this workflow is easy to use, robust, and provides an end-to-end solution for release glycan analysis, and can be used to improve productivity and also confidence in biotherapeutic development.